people. How are y'all? My name is Hector Corrada Bravo. I am at the Center for Bioinformatics and Computational Biology. I'm a colleague of Rob, who was here a few speakers ago, and Mihai, who I think was here a few speakers ago. And I'd like to give you a little bit of a sense of what my group works on. And kind of the general theme is interactive genomic data analysis. We work in biology and high throughput biology and the analysis of that data. I like to think of myself and my students wearing two hats. We try to make biological discovery from high throughput experiments, and in order to do that, we analyze data. We analyze large, messy, incomplete, hard to understand, but very important data. So that's one of the hats. The second hat is we make tools that make that job easier. So my motivation is making my life easier, and along the way, hopefully making the life of other people easier as well. And the way in which I think we are most effective when we're dealing with this kind of data is recognizing that data analysis is most effective when it is interactive. And what I mean by that is when there is tight integration of statistical, right, the way we infer things, computational, the way we compute the measurements and the inferences that we can make, and interactive visualization that allows us to, one, get a sense of the kinds of inferences that we would like to make, and a sense of what the data that was used to make those inferences is. So we've, for a while now, had a project, this is funded by NIH, where we build a computational statistical system for interactive visualization. It features tight integration of statistical analysis and computational analysis and interactive visualization. And the idea here is design visualizations that are most effective for each data type, incorporate the inferences that you might make from that data, be able to follow the trail of evidence from statistical inference down to the underlying data that was made, and be able to integrate publicly available data sets to understand what was happening there. So this is an interactive visualization that shows regions in the genome, right? We can think of DNA as this very long, one-dimensional domain. We make statistical inferences of interest that say there's something interesting happening there. And we say, well, why is that the case? Well, we use this data to make that inference. And if you think that seems to be important, you can study the underlying data, compare it to other underlying data that may also have an inference, and start to get a sense of, you know, the precision and sensitivity and robustness of your inferences. But of course, this is all tightly integrated to, you know, state-of-the-art statistical methods, some of which we borrow, many of which we also develop. Other thing that we're working on a lot is this ability of making that tighter by allowing steering of the visual exploration, which parts of the data, which parts of the domain, what measurements of the data, we have multivariate measurements, which measurements of and parts of the domain do you want to explore, and use statistical inference and computational methods to steer the user towards those methods, those areas, and make analysis interactive. And that opens up a bunch of interesting questions about how do you do discovery in this adaptive fashion? How do you ensure that as you're making discoveries sequentially, you're not making too many false discoveries? Statistical methods that kind of are very modern, trying to understand those problems. And as I said, our first hat and most important hat is to make biological statements, and the driving application here is in cancer epigenetics. These are modifications in DNA that are not encoded in DNA itself. So these are very susceptible to environmental changes, are involved in early cancer progression. So it's a viable place to start thinking about treatment and prevention and measurement of cancer. So that's one part of the questions that we ask. The other part of the questions that we ask are, right, suppose that we remove that nice organization of the genome as a linear space. How do we then explore data that has a more complex organization? So in particular, we're thinking how do you explore measurements and data in spaces that are not nicely organized? And the applications we have in mind, as Rob talked about, we are now measuring single cells. And he showed this plot that says as more data shows up, more data cell types appear. I would argue that's because we don't know what a cell type is. All right? And the reason we don't know what a cell type is because this kind of unknown combination between the measurement and the biology and what we know of biology. And part of the way we like to think of and where interactivity helps is this idea that similarity in cell type gives us a lot of information. So we can organize cells and cell types in those that are more similar to each other, those that are more dissimilar to each other, and construct a hierarchy of cell types to organize that search space. And then construct applications, interactive visualization applications that allows you to navigate that hierarchical structure and dynamically and adaptively look at the measurements we take. So for example, these are clusters of cells over a particular measurement, multiple measurements over time, and understand, as we do that interactively, possible patterns for what defines a cell type in different parts of that hierarchical organization. Other area that we work, as you might see, for interactivity, when we deal with data, both the measurement and the inference, is our ability to understand what models are doing. And for that, we spend a little bit of time as well thinking about applications where we try to understand and interpret what machine learning models are doing. This is not a genomic application that we have in mind. This is a neuroscience application that we have in mind. And again, this kind of came from a collaborator who has a measurement where they scan brain activity for individuals while they're performing a particular task. One example task is they show you a sequence of playing cards, and you have to remember what card they showed you 2 cards ago, right? While you're doing that. And we said, so that gives you a time series data for many, many locations in the brain. And we said, well, let's see, can we build some of these newer, or not newer, but deep learning models that are recurrent, so recurrent neural nets. And can we use some of these new spiffy interpretability and variable importance methods to say which parts of the brain seem to be important to predict what task the individual was doing at a given period of time. And we run that, and it says all the important parts of or the only time or the significant amount of importance for this task only happens at the end of the task. So everything that they were doing while looking at these cards come up and up and up was irrelevant. And we said, well, that doesn't sound right. Could be true. It doesn't sound right. So we said, all right, is there something about the properties of these networks that precludes the use of these new methods to understand how important these things are over time? And we show mathematically that saliency, this measure of importance, vanishes backwards in time. That's just the property of how these very, very, very often used methods behave. And that's not satisfying. So, in fact, with collaboration with Sohail, we came up with, first of all, an explanation as to why this is happening mathematically, and some newer models that are able to retain saliency across the entire task. We show by simulation that it's doing the right thing. Do we know for the neuroscience application that it's the right thing to do? We don't know yet. We're working on that. But at least we can say we could also do that using our newer methods, and we'll try to find out what the right thing to do is. But the combination between mathematically proving the behavior of these models and then showing how that may affect neuroscience is a lot of what we do. And the last little bit on how we attack interactivity is this idea of, Rob, breaking the pipelines that make data very, very, very efficient to process. All right, all these great pipelines that he's talking about are fantastic. They give us a lot of computational efficiency, but often we don't know exactly what they're doing, which is fine, but not satisfying. All right, so can we come up with what I call interactive statistics in such a way that we don't lose the computational efficiency of these modern pipelines, but produces measurements that we can interact with and understand when these pipelines create inferences, that we can go back and understand how they arrive at those differences, at those inferences. So the research group that I try to put together to build this kind of tools requires students who are interested and passionate in the data analysis and the inferences that we make from data analysis and how that affects biology and neuroscience, passionate about building systems, passionate about building statistical methods to derive those inferences. So we drive students from multiple areas in CS, cell biology, molecular genetics, applied stats, and math. We sit together often and collaborate with folks at the University of Maryland in Baltimore at the medical school, NIH, and others in CBCB. So if you'd like that idea of being interdisciplinary group, wear a few hats, come talk to me. If you do any of these things better than me, you're hired. If you do 2 of them, you're certainly hired. If you're 3, stay away. All right? So if you have questions, please come talk to me, and I'll be happy to address them. Thank you.